Good morning, dear students. Today we will study about the vestibule and cochlea and also how do we hear. Please listen to the class. Vestibule. Utriculus and sacculus are the two membranous structures located between the semicircular canal and cochlea of the inner ear. These structures comprises the vestibule. These structures are having a fluid in it, and that is called endolymph. And please, my dear students, one thing to be made very clear here, that is, in the inner ear, the fluid which is present in the inner ear is called perilymph with the semicircular canal, and that of the utriculus and sacculus it is called endolymph. The difference is that wherever this fluid is surrounded by bone, what is it called, bony labyrinth, bone, bony labyrinth, there we give the name perilymph. And wherever this fluid is surrounded by a membrane, membranous labyrinth, membrane, there we give name endolymph. But if a question come, name the fluid that is present in the inner ear, you will write only perilymph. You will not mention endolymph. Endolymph has to be mentioned only at certain places of the inner ear. One is utriculus and sacculus. The other one I will tell you in the course of time. These structures have the function of static balance of the body. So utriculus and sacculus have the function of static balance of the body. What is static balance? It is the balance of the body with respect to gravity. Balance of the body with respect to gravity is called a static balance. Now come to the cochlea. It is a 2.5 times twisted tube, looks like a snail's shell. It has two membranes in it which separate this tube into three different canals. So cochlea is a 2.5 times twisted tube, and this cochlea inside it, this tube is being separated into three different canals by two membranes. They are called this vestibular canal, median canal and tympanic canal. The membrane that separate the vestibular canal and median canal are called vestibular membrane. The membrane that separates the median canal and tympanic canal are called basilar membrane. This term is very important. Basilar membrane found between median canal and tympanic canal of the cochlea. The fluid present in the vestibular canal and tympanic canal is called perilymph, but the fluid present in the median canal is called endolymph. Here comes what I had mentioned earlier, that wherever the fluid, this fluid is surrounded by membrane, there we give the name endolymph, and wherever this fluid is surrounded by bone, there we give the name perilymph. So endolymph term comes in two different places. One is in utriculus and sacculus, and another is in the median canal or the middle canal of the cochlea. There also we name this fluid as endolymph. Otherwise, everywhere in the inner ear we give the name as perilymph. So if a question come, name the fluid present in the inner ear, you will simply write perilymph. But if it has been specially asked the fluid that is present in the utriculus and sacculus, you will name endolymph, or the fluid that is present in the median canal of the cochlea, here also the name is endolymph. In the median canal, on the basilar membrane has some sensory cells called organocorti. So location organocorti, it is found on the basilar membrane of the median canal of cochlea. So median canal of the cochlea has to be mentioned. Basilar, because basilar membrane has got two surfaces, one is facing the tympanic canal, the other one is facing the median canal. So organocorti is located on the basilar membrane of the median canal of the cochlea. These sensory cells, means organocorti, convert the sound vibrations into nerve impulses and transmit them to the auditory nerves. Auditory nerves transmit them to the temporal lobe of the brain, and we have the sensation of hearing. We can hear the sound frequency between 20 Hz and 20,000 Hz. Below 20 Hz we cannot hear, and above 20,000 Hz also we cannot hear. That is why sometimes, even though we find that there is no noise, but some animals like the cows or that of the dogs get very well means be attentive on different directions by looking at different directions. This means that they are capable of hearing such type of sound which is above,
can hear sound above
hertz, but
20,000 Hz.
Now please see here the cochlea and its cross section. At the right side, what you see, the cross section of the cochlea, and you can see all the three canals: vestibular canal, median canal and tympanic canal. Vestibular canal with perilymph, and tympanic canal also having perilymph, and median canal has endolymph. And also you can see the organocorti labeled on the basilar membrane of the median canal of the cochlea. Now, oval window and round window. It is a membranous structure located on the cochlea in front of the vestibular canal. This membrane is in contact with the states of the ear ossicle. Therefore, this membrane receives vibration from the states. So, when the states or the ear ossicle vibrates, then this vibration is carried into the inner ear through the oval window, because oval window also vibrates. The fluid inside the vestibular canal vibrates due to the vibration of the oval window. This vibration is sensitized by the organ of Corti. Round window is a membranous structure located on the cochlea just below the oval window in front of the tympanic canal, and it relieves the pressure exerted by the perilymph due to the vibration of the oval window. You know that the inner ear, the entire structure cochlea is made of bone, and if the perilymph of the vestibular canal is vibrating, then that vibration has to be carried out in the entire cochlea if there is a membrane just in front of the tympanic canal. Then only, if the tympanic canal does not have a membranous structure, only the bone is there over there, in that case the fluid present in that canal will not vibrate at all. So in order to relieve the pressure exerted by the vibration of the fluid present in the uh, canal, in the vestibular canal, as well as that of the tympanic canal, this round window helps in it. Okay, so in order to make it very clear, suppose if you have taken a PET bottle, a plastic bottle, and its upper side is cut off, then fill the plastic bottle with water completely, then cover the top of the bottle with a rubber bladder, then try to push the rubber bladder down. You will not be able to push it down because the bottle is already full of water. It cannot be pushed down. But if this experiment is performed by just cutting off the upper and lower part of the bottle, and on both the surface, upper side as well as the lower side, if you cover it with a rubber bladder, lower side you cover with a rubber bladder, then fill the bottle with water, then upper side also to be covered with a rubber bladder, then take the bottle in your hand and try to push down from one end, then you will be able to push it down because the other end has got rubber bladder. Okay, therefore you can easily push it down, or it can receive any kind of vibrations. Okay, the same is the role of the round window here too. The same is the role of round window. It relieves the pressure exerted by the perilymph due to the vibration of the oval window. You can see here the oval window is shown and round window is shown. Oval window is shown in front of the vestibular canal and round window is shown in front of the tympanic canal.
Now let us know how do we hear. The sound waves collected by the ear pinna is directed into the auditory canal, and these waves hit on the tympanum or the eardrum. As a result, the tympanum vibrates, and this vibration is carried into the middle ear through the ear ossicle. all the three bonds of the ear ossicles, all the three bonds of the ear ossicle vibrate, and they are arranged to an angle of 45 degree to each other, so they can amplify the sound vibrations. And the stem which is in contact with the oval window also vibrate. As a result, the oval window vibrate, and the fluid inside the cochlea also vibrate, due to which the sensory cells located on the basilar membrane of the median canal, that means organocorti, get sensitized by this vibration, and the organocorti generate impulses which is carried through the auditory nerve to the temporal lobe of the cerebral hemisphere, and we get the sensation of hearing. This is the way we can hear the sound frequency between 20 Hertz and 20,000 Hertz. Okay, above 20,000 Hertz we cannot hear and said to be supersonic sound, and below 20,000 below 20 Hertz also we cannot hear. This is the way we are hearing.
That's all for today. The rest we will study in the next class. Thank you. Have a nice day.